response. So the learning objectives of the lectures to describe the general events which happen during inflammation, to describe the pathological features that occur, to discuss two important components of this inflammatory response, namely the complement system, which you will get a more detailed lecture later, as well as the role of the mast cells in generating the symptoms of inflammation. To highlight to you the difference between this term that I'll be using of pro-inflammatory versus what is actually inflammatory. And some of the components can be interchangeable. In other words, some cytokines can be both pro-inflammatory and inflammatory. And then to discuss the important proteins called chemokines, which play a role in directing immune cells in the body. And then finally to talk about the effects of cytokines which are generated during an inflammatory response, their effects on the blood vessels, the endothelial lining the blood vessels. A lot of this material will be covered in more detail by pathology, by Dr. Braff. So, as I've said before, the inflammatory response is actually the war zone where the immune system is mobilized to combat an invasion by non-self. And in this scenario, you can imagine puncturing yourself with an infected splinter. And as these bacteria enter, penetrate the skin and enter the underlying tissue, you already have phagocytes in the form of neutrophils and macrophages there standing by to phagocytose. And as we said, they recognize these non-self cells via PRRs, which allows them to take them in. There are three main processes that are going on here. The first is that the system needs to block or plug the leak. And so that's brought about through the action of platelets, which will then bring about blood coagulation. The other is that you need mediators which will open up the vasculature to allow circulating cells and platelets to get out of the blood vessels into the tissue. So to recruit more phagocytes in the form of neutrophils, etc., to get into the site to deal with the invasion. All right? And a lot of this is mediated by this important system of blood proteins known as complement proteins. So the function of complement is one, that these proteins themselves, once they form the so-called membrane attack complex, can directly kill these bacterial cells. The other is that they can promote phagocytosis of the bacteria by macrophages and neutrophils. And the third is that they can trigger the symptoms of inflammation, redness, swelling, heat, etc., by their action on a type of cell known as the mast cell. And they recruit outside cells to that inflammatory site through the action of chemotaxis. Now, do you know what chemotaxis is? Have you heard of it? can also call them, yeah, so someone tell me what chemotaxis is. Right, chemical attraction. So the an example of this is, say you're walking past a bakery that's just baked fresh bread, and you can smell the pleasant aroma from that bread, right? And that attracts you to the bakery. And the closer you get to the bakery, the stronger the smell is. And so those are chemotactic factors, the aroma of the bread hitting your nasal senses. And the closer you get, the more concentrated those pheromones are. And that's basically how chemotaxis work. Now, in terms of one of the functions of complement to promote phagocytosis, there's an important term you need to know, which is opsonization. Opsonization refers to the facilitation of phagocytosis by a tag, for instance. So basically, an antibody is an opson. An antibody will bind to a bacterial cell if it's specific for it, and these phagocytes have receptors for the antibody. So the antibody acts like a mediator for phagocytosing the bacteria. We already know that these phagocytes have receptors already in form of PRRs for the bacterial cell, but these are additional means for these phagocytes to recognize and bind to these bacteria. And so, just like the antibody, these complement components can also act as opsonins. So in this diagram here, we have a bacterial cell that is bound by these complement proteins, tagged, and these phagocytes have the receptor for that complement. So then this bacterial cell bound with the complement gets attached to the phagocyte, which allows it to take it in. So this is another means of getting phagocytosis and binding. As I said, these complement proteins activate these other granular sites called mast cells, which are tissue-fixed cells usually located along blood vessels. They contain these granules so that when complement is activated, they will bind to the mast cell and cause it to release these granules. A major component of these granules is histamine, which pharmacology will give you a bigger lecture on later. And the effects of histamine is to cause vasodilation, in other words, to open up the endothelium of the blood vessels, allowing for more leakage of plasma fluid in the vessels and cells to get into the underlying tissue. The mast cell also releases prostaglandins, which also aid in this, and inflammatory cytokines. We normally associate mast cells with allergies, type 1 hypersensitivity. This is why you get sneezing, runny nose, teary eyes. A lot of that is due to mast cells. But these mast cells act normally in an inflammatory response. Then we have these cytokines produced by these activated macrophages and dendritic cells. I've got 6 listed here. These will be repeated throughout this course. So first of all, there are 3 major inflammatory cytokines, which are interleukin-1, which we talked about in the last lecture, the inflammasome. interleukin-6, and human necrosis factor alpha. These have a wide range of effects on all different types of cells. One is something like TNF-alpha, which is called human necrosis factor because it was originally identified as being able to kill cancer cells. So this is a protein which just binds the so-called death receptor on tumor cells, causing it to undergo apoptosis. So it's directly cytotoxic. And in a similar manner, it can kill bacterial cells. These three cytokines are inflammatory in that they also cause vasodilation, but also have effects on the endothelial cells in terms of stimulating them to express so-called adhesion molecules. Then we have a chemokine, CXCL8. I'll tell you why it has the name CXCL, what it means later. Another name for this chemokine is IL-8, because when it was originally identified, it was thought that it was a cytokine, but subsequent studies showed that it was actually a chemokine. And then we have two important, what we call pro-inflammatory cytokines. Pro-inflammatory are cytokines which stimulate cells like macrophages to produce inflammatory cytokines. So they kickstart the inflammatory process. And these two are interleukin 12, which is mainly produced by macrophages, which stimulate natural killer cells to produce the inflammatory cytokines. And this important one, which you'll hear a lot more about later on, interferon gamma. So interleukin 1, interleukin 6, and TNF-alpha activate the acute phase response. These acute phase proteins are produced in the liver during an inflammatory response. So they are used as biomarkers to determine if the patient has inflammation or not. They have a wide range of effects. One of them is that they cause fever, a rise in temperature. The basis for this is that if you have a rise in temperature in the body, that makes it more hostile for bacteria to grow or any pathogens. These acute phase proteins are also consist of these complement components. So you get more of these complement proteins being generated during an infection. And then also these clotting factors to plug the leak. As I said, when these inflammatory cytokines are produced within that tissue, they'll get released by the macrophages, and these cytokines will diffuse towards the blood vessels. The endothelial cells have receptors for these cytokines, and once these cytokines bind to these receptors, they will stimulate the endothelial cells to express these so-called adhesion molecules. Now imagine a blood vessel where the blood pressure is so high that it's almost like a raging river. And you've got these leukocytes circulating through the blood. And now they have to stop in the face of this pressure, blood pressure, to get through the blood vessels into the underlying tissue where the infection is. So the function of these adhesion molecules is to act as anchors so that these circulating leukocytes can stick to these adhesion molecules in the face of the blood rushing. The cytokines act, as I said, to cause vasodilation. So it means that the endothelial wall opens up. And so with this tethered leukocyte, it then allows it to gradually work its way along and then squeeze itself through the endothelium to get to the underlying tissue where the infection is. This process is called extravasation. And this process of squeezing through the endothelium is known as diaphoresis. But Dr. Grau will tell you more about this later in the pathology lecture. So this is just a cartoon showing this process. So now these chemoattractants called chemokines, they are small proteins, and there are three types: CC, CXD, C, or four kinds, and CX3C. C stands for the amino acid cysteine. What do you remember about cysteine? Causes disulfide bonds. So basically, these four species, based on their cysteine residues, determines the shape of the chemokine. So we have CC, which looks like a G. No, this is a single C. This is CC, so it's two disulfide bonds. CXC, X is a convention in biochemistry where X is any amino acid. Right? I don't know if you learned this in biochemistry, that you have an alphabet for each amino acid. Can't remember. R is arginine. That's all I remember. But basically, X is any amino acid. So we have the CXC chemokine and the CX3, any amino acid in between two systems. Don't worry too much about these details, but just be aware of this is how the convention is. So, okay, now this is where it gets complicated, because by convention, chemokines work like everything in terms of a ligand and a receptor. So when we talk about this extravasation, for instance, these leukocytes might be expressing CXCL, the ligand, being attracted to a chemokine CXCR, which is present on here. But it's interchangeable. Depending on the situation, this might be the other way around. So the R, the receptor, by convention, might be named on the stimulated cell. The ligand might be present on the other cell. Right? It's complicated. Now, two of the major chemokines attract neutrophils from the bone marrow. And this is important because during an inflammatory response, as I said, the bulk of the phagocytic cells there are neutrophils. So these activated macrophages and so on, releasing these chemokines, as well as this other cytokine, which you'll learn later, is leukin 17, stimulates proliferation of neutrophils within the bone marrow and draws them into this infected site. So basically, this is what we have then, that if we have a local infection in tissue, these activated macrophages, either through the production of the inflammasome, the activation of the PRRs, opsonization, etc., release these inflammatory cytokines and chemokines, which will promote generation or proliferation of leukocytes in the bone marrow in the case of neutrophils, as well as releasing these chemokines to attack them to that tissue site, as well as stimulating the expression of adhesion molecules on the endothelium to allow for this passage through the blood vessels. So this is just basically a summary of the three major or four major, I can't count, five major mediators in this inflammatory. And again, this will be talked about in a lot more detail from pathology. So we have first and foremost complement, which activates mast cells to produce histamine. Also have these kinins, which also cause vasodilation and also activate pain receptors as part of the inflammatory response. The lipid mediators, prostaglandins and leukotrienes, which again we normally associate with type 1 or allergic response. But just be aware that these processes don't just happen in allergies, but also in a normal inflammatory process. And then these cytokines, TNF, IL-1, IL-2. Now, once the infection has been dealt with, so once all the bacteria have been phagocytosed or killed, you need a mechanism to stop that inflammatory process. You need to block complement proteins. You need to stop the production of these inflammatory cytokines. Because if you don't, you're going to get chronic inflammation, which again, pathology will tell you more about because that can cause serious disease. And so you have several mechanisms for this downregulation. One is that you have a specialized type of lymphocyte called a regulatory T cell, which I'll talk about in another lecture, whose major cytokines are interleukin 10 and TGF-beta. And these are immunosuppressive. So they will block any inflammatory T cell from making any of these inflammatory cytokines. The other is that there's a type of macrophage which releases antagonists for the inflammatory cytokines like IL-1. So in other words, this is like, say, you can think of it as a soluble receptor that will bind to IL-1 as it's circulating in the blood, preventing that IL-1 from binding to a macrophage or whatever to generate the inflammatory response. Then you have other factors which bind to complement protein and will again prevent those from binding to receptors or being activated further in the complement process. And then there's this is very new, but we've now discovered a whole set of proteins called resolvins, which actually resolve the inflammatory process. We know very little about these. They seem to be lipid-based molecules. And then finally, once you've blocked this inflammatory process, you need to regenerate and repair the damaged tissue caused by the killing created by complement, etc. One of these is this cytokine TGF-beta, which promotes wound healing and fibrosis. Again, I will talk more about this later on. Okay, that's quite a short lecture. Sorry? Yeah, something like this. Okay.